Welcome to the second topic of Chapter 13. Here are our objectives for the chapter. If you have any questions or need help on a topic, please let me know. For the most part, I hope this chapter and Chapter 14 are as much of a review for you as Chapters 1, 2, and 3 were. Now that we've talked about glycolysis, we can move on to the citric acid cycle, biosynthesis, and the regulation of metabolism and nutrient storage. Like with glycolysis, I want you to memorize the structures of the intermediates of the acetyl-CoA transformation, the citric acid cycle, and the names of the enzymes. The second process that we're going to talk about in this chapter is the citric acid cycle. Before we discuss the citric acid cycle, we need to talk about acetyl-CoA transformation. This is the process where pyruvate is transformed into acetyl-CoA, generating NADH and CO2 as a byproduct. This transformation is accomplished by the pyruvate dehydrogenase complex. This creates a product that can easily enter the citric acid cycle. Pyruvate isn't the only molecule that can go through this transformation. Some fats and proteins can also enter the cellular respiration process through this acetyl-CoA transformation. Now that we've transformed pyruvate into acetyl-CoA, let's take a look at the citric acid cycle. This is the last step in the breakdown of food. In this stage, we remove the electrons from the acetyl-CoA and shuttle these via high-energy electron carriers, NADH and FADH2, to the electron transport chain, and while oxygen is not directly a part of the process, oxygen presence is required for this process to run. This is because the NADH and FADH2 molecules are recycled in the electron transport chain, or ETC, which needs oxygen to function. If these molecules are not recycled by the ETC, the citric acid cycle will stop functioning. Don't forget about the CO2 either. This gas is what we breathe out and is the main byproduct of the citric acid cycle. Let's spend some time reviewing the 8 steps of the citric acid cycle. In step 1, acetyl-CoA enters the cycle and meets the oxaloacetate intermediate that has been recycled from the previous spin on the cycle. In this stage, these two carbon molecules form one molecule, citrate, and coenzyme A is removed. This coenzyme A is recycled so that it can be used in acetyl-CoA transformation again. In step 2, we again do a molecule shuffle or isomerization, rearranging the hydroxyl groups on the carbon molecules. This results in an isocitrate molecule. This is an energetically more favorable conformation to lose the electrons to NAD+ in step 3. This oxidation in step 3 is the first of 4 that will occur in the remaining steps of the cycle. This particular oxidation sends one NADH molecule and a proton to the electron transport chain. We also see our first CO2 byproduct created in this step when an acid group is snipped off. In step 4, we see the return of coenzyme A. In this stage, CoA is added to the carbon molecule through a process very similar to acetyl-CoA transformation. The acid group next to the ketone is snipped off and is released as CO2. Through this process, we see our next oxidation when NAD+ picks up a hydrogen as coenzyme A attaches. We don't want to lose the energy in that hydrogen. Step 5 is a multi-step process where water enters the system followed by a phosphate group. This addition of the phosphate group removes coenzyme A from the molecule. However, within this step, the phosphate is removed to GDP, forming GTP. GTP is very similar to ATP, only instead of adenine, the molecule carries a guanine. In step 6, we begin the transition back toward oxaloacetate through an oxidation step that actually removes 2 electrons and protons and forms NADH2. Step 7 sends our molecule through a rearrangement again in preparation for step 8. In step 8, the molecule undergoes the last oxidation step, creating one last NADH molecule and recreating oxaloacetate, ready to process the next acetyl-CoA entering the cycle. So let's review the important aspects of the citric acid cycle. Remember, in the citric acid cycle, 3 NADH and 1 FADH2 are produced for each turn of the cycle, and each glucose molecule that enters glycolysis causes 2 turns in the cycle. The main byproduct of the cycle is CO2, which exits our body through our breath. As well as learning the intermediates and the enzymes of the citric acid cycle, be aware of the basic concept for each step so that you can explain what the molecule goes through to create these high-energy molecules. As we have been talking about, the high-energy electrons go to the electron transport chain for high amounts of ATP generation. This is the focus of Chapter 14, so we won't go into too much detail right now, but I wanted you to see that the NADHs and FADH2s that are produced in glycolysis, acetyl-CoA transformation, and the citric acid cycle are used to power the oxidative phosphorylation of the electron transport chain. One more note before we go into the control of these processes. We always think about glycolysis and the citric acid cycle being responsible for participating in energy generation, but they also play a role in biosynthesis. At various stages in the cycle can be stopped and intermediates harvested for biosynthesis of organic molecules within the cell. We have talked about this many times, but the cell would be lost without regulation controls. As you can see from this slide, the feedback mechanism is very intricate and complicated. This allows the cell a great amount of finesse when controlling catabolism and anabolism. In this last section of lecture, we are going to talk about some of these control mechanisms. Before we move too far, let's remind ourselves of the ABCDs of metabolism. Anabolic systems build molecules, while catabolic systems destroy molecules, or break them down into smaller pieces. Let's take a look at one example of these feedback regulation cycles. This is the relationship between glycolysis and gluconeogenesis. Gluconeogenesis is the process of making glucose out of other biological molecules. This is especially helpful during times when food is not readily available or when the body is undergoing strenuous physical exercise. The blood glucose level is extremely low. These two systems are effectively controlled in 3 places in the glycolysis cycles: steps 1, 3, and 10. These are irreversible steps in the process and are easy places to turn on or off when energy demand is either low or high. To get around these irreversible steps, gluconeogenesis has its own alternative steps that you can see here on this slide. An example of this regulation control can be seen at step 3. When the cell is low on ATP, the enzyme phosphofructokinase is activated, pushing the glucose intermediates through glycolysis to create ATP through chemiosmosis. Conversely, when ATP and other products are plentiful, this enzyme is turned off, pushing the reaction towards gluconeogenesis and towards storage of food products. Let's wrap up this chapter by looking at how cells can store these food products. Looking at this, you will note that gluconeogenesis requires quite a bit of energy. This isn't a surprise considering that this process is going against the laws of thermodynamics. Obviously, this is not an ideal process for the cell to undergo regularly. Instead, the cell uses storage to keep a small supply of backup nutrients available. Our main short-term storage of glucose is in the form of glycogen and mainly stored in our muscles and liver. This storage of glycogen is used at night when we are sleeping or for the first bit of sustained exercise, about 15 minutes. This is an easily accessible nutrient for the cells. The generation of glycogen is allosterically regulated by the presence of glyceraldehyde 3-phosphate, or G3P. When G3P and ATP are present in the cell, the enzyme glycogen phosphorylase is inhibited. This prevents the further breakdown of glycogen when there is enough energy already present in the cell. When the phosphorylase is inhibited, a second enzyme is stimulated, glycogen synthase. This will use excess G3P to create stores of glycogen. The body does not only store glucose as glycogen, it also stores fat. Fat storage is very important to the cell because the breakdown of 1 gram of fat releases twice as much energy as the breakdown of 1 gram of carbohydrates. We can store these fat molecules as droplets in the adipose tissue of the body. These stores are used for long-term energy needs when food is not present. One example of this is after a night of not eating, the body will have depleted all the glycogen stores and must break down fat to keep the cells going when you haven't eaten in a while. One last note on this storage. Plants store nutrients slightly differently than we do. For example, they store excess glucose as starch instead of glycogen. Examples of these are potatoes. Plants also store fats and starches in the chloroplast so that they may be broken down when light is not available to create ATP for the Calvin cycle. We will talk more about this in the next chapter. Plants also pack these nutrients to be used by seeds as they grow into new little plants. You can see some of the examples on this slide. This is the end of chapter 13. Hopefully most of this was review for you. Take a moment and review the objectives and make sure that you are as comfortable as possible with this before continuing on to chapter 14. And of course, let me know if you have any questions. I'm happy to go over these topics with you until you understand them.